Welcome to the second video. This video is going to focus on both transcription and translation. It actually will end up being probably cut into two videos just to sort of not have me have to stop in the middle. So in transcription, like we discussed on the first slide, DNA is transcribed into RNA. We are going to now be looking at a lot of the details here, but again, you need to remember the big picture, okay? That you start with the DNA, okay, and the DNA contains letters, which we'll see how in a second, or nucleotides, A, T, G's, and C's, that code for a protein by calling for specific amino acids to be put into the protein. We are going to be looking at the big picture of how physically that's done. But again, you need to remember the big picture that the nucleotides in DNA code for the amino acids that are going to be inserted into a protein, for a protein that's going to do a job in the body. But we're now going to sort of look at the nitty-gritty details of how that's done. So first of all, there are two strands of DNA. There's one strand of DNA which is called the template strand, okay, and that's the strand that is copied into RNA. So down here, this one here is the all green, or this molecule is the mRNA. This here is the template strand, because it's the one that's serving as the template to copy the mRNA. Then the one that is not being transcribed is known as the coding DNA, or the coding strand. It has the same letters, except that T is T instead of U for the mRNA, the same nucleotides, I always want to say letters, as the mRNA that's being made. Okay. The enzyme that copies the DNA into RNA is known as RNA polymerase. And similarly to replication, the DNA needs to be unwound, and it adds the corresponding correct mRNA bases to the DNA. It also occurs, as the PowerPoint has just informed us, on a 5' to 3' direction, just like DNA replication. Now, there are many genes in the DNA, so the question is, how does the DNA know which gene it wants? Well, all genes in front of them have something called a promoter region, and inside of the promoter region is something known as the Tata box. Now, how the cell or the DNA knows which gene to transcribe is that genes have specific transcription factors. Okay, and these transcription factors bind to the Tata box, or the promoter. And what they do is they promote the binding of mRNA to that gene. If you remember when we were learning about cell communication, we said how sometimes the response is that a transcription factor goes into the nucleus and causes the production of a protein. Well, this is how it happens. Specific transcription factors bind to specific regions of DNA, and they can only bind to those regions of DNA. And then when those transcription factors are bound to the promoter region of the DNA, they recruit RNA polymerase to the DNA so it knows to begin transcribing the gene. So again, this whole thing about transcription factors is important, and we're actually going to get more into this whole idea of transcription factors right before Christmas break. But to start transcription, it's usually a group of transcription factors bind to the promoter region of DNA. And that recruits the binding of RNA polymerase to the DNA, which essentially either turns on transcription by bringing RNA polymerase to the DNA, or if the transcription factors are not there, turns off transcription because RNA polymerase cannot be brought to the DNA. Once RNA polymerase is brought to the DNA by transcription factors, it is the job of the enzyme RNA polymerase to add the matching RNA bases to the template strand of the DNA. And again, this proceeds in a 5' to 3' direction, just like the DNA application. Okay, now, this idea is new, something that we never discussed as freshmen, is that inside genes, okay, there are areas of the DNA that actually code for the protein, and areas of DNA that don't. The areas of the part that's being transcribed that code for the protein are called exons. Okay, and these are the expressed or that's why it starts with EX, regions of the DNA that code for the protein. Then, within the mRNA that's made, we also have introns. These introns are known as the junk DNA. They don't code for proteins. And what we're going to see in the next slide is that these introns need to be cut out of the DNA. Now, if you look here, and this is DNA, so DNA, the introns are both in the DNA and the mRNA. You'll see here that the blue sequences, okay, those are the exons, the part of the DNA that codes for the protein, and the orange sequences here are the introns, or the junk DNA, which, when this is transcribed into mRNA, is going to need to be cut out. Now, the process of cutting out introns from mRNA is known as mRNA splicing. Okay, if you look down here, this is the DNA, and the DNA is transcribed by mRNA polymerase directly into the mRNA. What's different here is that this is the RNA bases with U, and these are the DNA bases. Then what happens is that we're going to see how in a second, is the exons, in this case, which is the orange DNA, are cut out, or the orange mRNA, or orange DNA up here, are cut out, and the green, which are the exons, are spliced together. Here, before the mRNA is spliced, this is called pre-mRNA. Then the introns, the green pieces here, are cut out and spliced together, and this is called the mature mRNA. As we bring the labels back. Now, it is important that splicing be accurate. If we look down at the image at the bottom, you'll see splicing one letter incorrectly changes the reading frame and changes the whole amino acid. If you take a look here, the difference between, so the red is the intron, and if you take a look at the difference in splicing, here we have an extra G that was put in compared to here. That incorrect splicing, okay, causes a completely different protein to be made. In fact, the different splicing causes the protein to stop early, or the protein not to be completely made, or first of all, it changes the amino acid here, these serine to valine and asp to arg, but then it actually causes the protein to be truncated or smaller than the one that should be produced. So all that being said, again, it's important that splicing be accurate. Being off by one letter, so here you have an extra G in the DNA here because we splice one extra letter here, causes the production of an entirely different protein. So it's important that the splicing be accurate. The name of the protein that does the splicing of the introns out is known as the spliceosome. The details of how this used to happen need to be known for the AP exam, but the spliceosome is made up of many small nuclear RNA molecules and proteins that combine the spliceosome complex. And the spliceosome complex binds to a sequence of DNA that it recognizes on either end of the intron. And again, how they recognize these sequences are not greatly known. And they form this lariat with the intron, and then the intron gets cut out. Again, the details of this, which used to need to be known for the AP exam, no longer need to be known. Really, what all that needs to be known is that spliceosomes cut out introns for the AP exam. Now, some of you may say, well, why have introns? What's the point? And the point is, and again, this is also not well known how it does it, depending upon the conditions in the cell, different parts of the DNA serve as introns sometimes, and other times they're exons. So if you look at this DNA here, there are sort of three different types of, or three exons that, or five, I guess, different exons that we have. Depending upon the conditions in the cell, this mRNA can be spliced differently. Okay, it can be spliced so that the green exon is an exon and included in the DNA, or it can be spliced so that, in this case, the green is cut out, and in this case, the red is an exon, and the green is an intron. This allows a number of different proteins to be made from the same mRNA. That idea is important. I'll repeat it. because mRNA can be spliced differently, or different introns can be cut out, that allows you to make more than one protein from one mRNA molecule. That is important, and the AP exam loves that idea. The idea that if you splice an mRNA differently, which does happen all the time, what's not really well understood is how the cell knows when to splice it one way or another. You'll get a different protein, or can get different proteins from one mRNA molecule. Once the mRNA is spliced by spliceosome, there are two more things that need to happen to it before it can leave the nucleus, so it's protected from enzymes that like to break down mRNA in the cytoplasm. First, a GTP cap is added, which is essentially a guanidine triphosphate molecule that's added to the cap, the 5' end of the molecule. This serves two purposes. It does help protect the molecule, and it also actually helps transport out of the nucleus. This allows the nuclear pore to hold on to it while it's being transported out of the nucleus. But it essentially ends, so it's a guanosine triphosphate molecule to the 5' end of the molecule, which is essentially like an ATP molecule, except that it has the nitrogen-based guanine instead of adenine at the top. The second thing that's added at the 3' end of the DNA is a poly-A tail. The sole purpose of the poly-A tail, which is lots and lots of A nucleotides, is to protect the mRNA molecule from degradation by enzymes in the cytoplasm. It has been seen that the longer this poly-A tail is, the longer the mRNA lasts in the cytoplasm, and the more protein it produces. But over time, what happens if this poly-A tail gets short is that the mRNA will be degraded by the cell. So the poly-A tail totally prevents degradation of the mRNA by enzymes in the cytoplasm. Okay, now this is going to be difficult for me because, again, I'm not on a smartboard. I'm laying actually in my dad's bed, guest room, doing this. So we need to transcribe this DNA template into RNA. The intron here is what is underlined. I don't know why it is bolded. So to transcribe this into DNA, all the letters are initially transcribed by mRNA polymerase. So I'm going to pause the video and do it because I'm running out of time, and then you can transcribe it while I'm pausing the video, and we'll see if we turn out with the correct thing, which is this. Again, this is not that hard. Okay, so I got tired of writing and just decided to type it. So if we take a look here, this is our mRNA that would be made from that DNA. Be careful that whenever there's an A in DNA, that A is transcribed to a U, not a T, because there is no T's in DNA and RNA. Then, again, this is the pre-mRNA. What's going to happen then, the spliceosome is going to come and cut out this segment right here. So let me get my pen. Pointer options pen. So here, this segment of DNA, or sorry, RNA, not the DNA never gets cut out. It's going to be cut out by spliceosome. And the two ends, which are called exons, will be spliced together to yield that molecule. This here is the mature mRNA. Hopefully I didn't make any mistakes by typing. It's possible that I did. Okay, so now, in the next video, which I have to split this video into, we're going to be talking about translation. So I would suggest going to answer the guided questions that we have answered up to this point, and then coming back to view the new link about translation.